Inside every single simple life form, we're talking the bacteria in your gut, the microbes in the soil, there's this fundamental story playing out millions of times a second. It's the story of how life reads its own instruction manual. So today, we're going to decode that process, prokaryotic transcription. It all kicks off with this really big puzzle. Think about it. A single bacterium like E. coli has thousands of genes packed into its DNA, but it's definitely not using all of them at once. So how on earth does it know which genes to turn on to digest your lunch versus which ones to fire up for self-defense? That's the mystery we're solving today.
And the answer? It's a process called transcription. Honestly, the best way to picture it is like this. Imagine the cell's DNA is this huge, priceless master cookbook. It's got every single recipe the cell could ever possibly need. But you're not going to drag that giant book over to the stove, right? Of course not. You just copy the one recipe you need onto a little recipe card. That's literally what transcription does. It copies a single gene, one recipe, from the DNA onto a portable message, a little recipe card, called messenger RNA, or mRNA.
Okay, so to pull this whole thing off, the cell uses a really specialized molecular toolkit. Let's meet the key players in this little microscopic drama, the scribe and the cookbook.
First up, the DNA itself. Now, we all know DNA is a double helix, right? It looks like a twisted ladder. But for transcription, the cell only reads one side of that ladder. That side is called the template strand. It's the master copy, the one with the exact instructions for the recipe it's copying. But wait, with thousands of genes all lined up, how does the machinery know where one recipe starts and the next one begins? Well, that's the job of the promoter. It's a special sequence of DNA right before a gene that basically acts like a giant flashing neon start here sign. It's what flags down all the transcription machinery.
And the machinery that sees that sign and pulls over? That's the star of our show, RNA polymerase. This enzyme is the molecular scribe. It's the workhorse. Once it finds that promoter, it locks onto the DNA and just starts building that new mRNA strand, reading the template and adding the right building blocks one after another.
But here's the catch. RNA polymerase is amazing at building, but is actually not so great at finding that start here sign all on its own. It needs a guide. So it teams up with this special helper protein called a sigma factor. And together, they form the complete package, which we call the holoenzyme. Think of the sigma factor as a molecular GPS that guides the polymerase right to the promoter, making sure transcription starts in exactly the right spot.
So we know that template strand gets read. But what about the other strand of the DNA ladder, the non-template strand? Well, here's a pretty cool detail. This other strand is almost a perfect copy of the final mRNA message that gets made. The only tiny difference is that DNA uses the letter T, where RNA uses the letter U. It's a little quirk, but it makes it a super useful reference for scientists.
All right, so we've met our cast of characters. Now let's see them in action. The entire process of transcription really does unfold like a nice, clean 3-act play.
Act 1 is initiation. This is where it all kicks off. The full holoenzyme, guided by its trusty sigma factor, locks on to the promoter. Then the DNA unwinds just a little bit, creating what we call a transcription bubble. Act 2, elongation. Now we're cooking. The sigma factor pops off, and the core RNA polymerase enzyme just starts chugging along the DNA, building that new mRNA strand at a crazy fast pace, about 40 nucleotides every second. And finally, Act 3 is termination. The polymerase hits the end of the gene. It lets go of the DNA, and it releases our brand new, shiny, completed mRNA recipe card.
Okay, this brings up a really important question. Starting in the right place is critical, but stopping in the right place is just as important. You don't want your recipe to be too long or too short. So how does our little molecular scribe know when the recipe is finished? Well, prokaryotes have come up with 2 really clever ways to do this. The first one is called row-dependent termination. And it's pretty wild. There's this protein called Rho that actually latches onto the new mRNA and starts chasing the polymerase down the strand. When the polymerase pauses at the end of the gene, Rho catches up, collides with it, and basically just knocks it right off the DNA. The second method, Rho-independent, is even cooler because it doesn't need an extra protein. Instead, a specific sequence that gets copied into the mRNA folds back on itself, forming this super tight little structure called a hairpin. And that hairpin physically pries the polymerase right off the DNA. How smart is that?
So we get how genes are transcribed. But this brings us all the way back to our very first question. Which genes get transcribed and when? This, my friends, is where a whole other crucial layer of control comes into play.
And this control is handled by a class of proteins called transcription factors, or TFs for short. These guys are the real decision makers. They're basically molecular light switches. They can bind to the DNA, usually near a promoter, and they can either help RNA polymerase get started or totally block it. They are the gatekeepers of the entire genome.
And if we take a look at a classic bacterium like E. coli, the breakdown of these TFs is really fascinating. About 43% of them are repressors. Their job is to turn genes off. Around 35% are activators. They turn genes on. And then you've got this really versatile 22% that can actually do both, depending on what the cell needs at that moment.
And their jobs are so elegantly simple, but so powerful. Activators are like a helpful guide. They make it way easier for RNA polymerase to find and grab onto the promoter, which effectively turns the gene on. Repressors, on the other hand, are like a security guard standing in front of a door. They physically bind to the DNA and block RNA polymerase, preventing it from ever starting. They turn the gene off. Simple as that.
So why go through all this trouble? Why have this complicated system of on-off switches? Well, the point here is that this isn't just about being efficient. For a lot of these organisms, this is a matter of life and death.
7. Just 7. What on earth could this number mean in the huge, complex, genetic world of a bacteria?
Well, get this. It means that in E. coli, an incredibly small number of transcription factors, just 7 of them, called global regulators, act as master switches that control the expression of over half of the cell's entire library of genes. It's an absolutely mind-blowing level of control, where just a few key decision makers can coordinate massive changes across the whole cell.
And this is where that control becomes, well, a weapon. For pathogenic bacteria, these switches are how they attack. They have specialized transcription factors that are basically sensors. When they infect you, these sensors are constantly scanning your body for changes: a shift in temperature, a change in pH, the amount of iron or oxygen available. And when they detect the right signals, they flip the switch, activating all the genes for virulence, and the attack begins.
And this just shows how absolutely vital these switches are. Researchers Zhou and Young put it perfectly when they said that if you disrupt these key transcription factors, the pathogen's ability to cause disease just plummets. In other words, without their managers calling the shots, the bacterium's response is a complete mess. Its whole attack plan just falls into disarray. So when you peel it all back, from a simple little recipe card to this incredibly complex network of genetic switches controlling life and death, prokaryotic transcription is just a masterpiece of biological engineering. Understanding how these circuits work is absolutely key to everything from fighting diseases to designing new biotech. Which of course leaves us with the ultimate question for the future: what amazing new capabilities will we choose to turn on next?